Welcome to the Development in Snails module. In this talk, we're going to learn about how cleavage occurs in snails. Snails are protostomes and they belong to the phylum Mollusca. Now, snails do have shells, but not all mollusks have shells. When we look at the phylum Mollusca, there are three main classes. The first one are the cephalopods that have the squid and octopuses as members of this class. They normally do not have shells. The next class is bivalvia, where we can see mussels, clams, or scallops. Oysters also come under the class of bivalvia, and these usually have shells. The class of gastropoda, or the gastropods, comprise of snails, slugs, conches, and other types of organisms. Now, snails have been traditionally used as a model to study development because they're abundantly found. They grow well in a laboratory, so they're low maintenance. They show variations in development that can be related to environmental cues, and they have large eggs that can be observed easily. The eggs develop rapidly, and so they don't take too long to develop. And thus, snails are good models for studying autonomous development too, because as we see, during embryonic development, autonomous specification plays an important role. Thus, the snails are good models. Now, all the examples that have been listed here can also be applied to other organisms that have been used to study development. Now let us observe what happens during the cleavage process in snails. Now this occurs after fertilization, and snails show a spiral holoblastic cleavage. Now an easy way to remember that snails undergo spiral cleavage is if you just remember how their shells look. Their shells look spirals, and thus that way it's easy to remember that the cleavage in snails shows spiral holoblastic cleavage. Now in the case of a spiral cleavage, the cleavage planes are not parallel or perpendicular to the animal-vegetal axis. Cleavage actually occurs at oblique angles. Now let us see what this actually means in the context of the figure that is shown below. In this case, the zygote has already gone through one cell division, and so we can see two cells, which are the AB and the CD cells. The top part is the animal pole, whereas the bottom part is the vegetal pole. Now, if we focus on the CD cell, which is shown in the form of that black circle, we can see that it is going to undergo mitosis and a spindle has been formed. Now, if you look at the plane of which the spindle is formed, it is not on that vertical axis, so it is not parallel to the animal-vegetal axis. Neither is it perpendicular to the animal-vegetal pole or axis. It actually happens in a plane that is at an oblique angle, or it is at an angle to that animal-vegetal axis. Now when this cleavage happens, we end up with the four-cell stage. So the first two cell divisions result in four blastomeres. Now, depending on the species of snail, these four blastomeres can be the same size or they can be different sizes, but in all cases they are called as macromeres. Now, when the next set of cell division happens, now we can see if we focus on the one capital A and the one small a, this is where the A macromere has undergone cell division. And we can see that the one capital A and the one small a, they're not on top of each other, but they're at an angle to each other. And this is what we mean by a spiral cleavage. Now, after the four-cell stage, each successive cleavage results in the macromere undergoing cell division to form a cell that remains as a macromere and another cell that forms a micromere. Thus, when we look at the picture to the extreme left, when we see the one small a, that refers to the micromere that arose from the one capital A, which is the macromere. Now, each successive micromere is displaced to the right or to the left of the sister macromere, and this is what provides a spiral pattern. The reason the micromeres are to the right or the left of the sister macromeres is simply because cleavage occurs at oblique angles, and thus gives a spiral pattern. And that is what is shown in the figure. So in the middle panel of the figure, we can see that the macromere has undergone the second cell division, and this is where we see the 2A has given rise to a small 2A, and the one that was the 1A that came during the first cell division of the macromere has formed two daughter cells, which are 1A2 and 1A1. And now if you look at the picture that is to the extreme right, we can see another cell division has occurred, which is the third cell division, where the macromere, which is now 3A, formed a micromere, which is 3 small a. Now if you follow all the different cells that arose from that macromere, where we see numbers associated with the alphabet A, we can see that they all form a spiral pattern. Basically, we're trying to say is they're not right one on top of each other in a vertical line or in a horizontal line. They form a spiral, and this is the hallmark of spiral cleavage. We all know that snails have shells, and when we look at shells, they show these spiral structures. Now, these spiral structures can be left-handed or right-handed. Similarly, snails can have their coils opening to the right of the shell, which is called as dextral coiling, and those kinds of shells are shown in the figure right here. Or the snails can have their coils opening to the left of the shells, which is called sinistral coiling, and that is shown in the figure that is to the left. Now the direction of coiling, so whether it is dextral or sinistral, depends on the orientation of the cleavage plane during embryonic development. We now know that the direction of the snail shell coiling is controlled by a gene called as the Formin gene, and there are two such alleles of the Formin gene. The dominant allele is given by the letter capital D, and the recessive allele is given by the letter small d. Now, depending on the combination of the capital D and small d alleles, the shell coiling can be dextral or sinistral. So, the capital D, and capital D small d genotypes give rise to dextral coiling, while the small d genotype gives rise to sinistral coiling. It should be noted that dextral coiling can also be called as right coiling, and sinistral coiling can also be called as left coiling. Now, the direction of shell coiling development in the embryo is dependent on the genotype of the mother. So it does not depend on the genotype of the embryo, it actually depends on the genotype of the mother. And this is called as maternal effect. So let's look at an example where we have a female snail that has the genotype capital D, mates with the male snail that has the genotype of small d. The resulting offspring will all have the genotype of capital D small d. Now, in general, capital D small d should provide a phenotype of dextral coiling or right coiling. And in this case, this is what is actually observed. Now, let us take an example where we switch the genotypes of the male and the female parents. So the male has a genotype of capital D, while the female has a genotype of small d. Once again, in this case, all the offspring will have the genotype of capital D small d, and we should expect that they all will again show dextral coiling or right coiling. However, surprisingly, they don't. They actually all show left coiling or sinistral coiling. And the explanation for this is that in this scenario, when we look at the genotype of the mother is a homozygous recessive genotype, which is the small d. And small d corresponds to the sinistral coiling, And that is why all the offspring are showing sinistral coiling or left coiling, because the mother has a genotype that corresponds to sinistral coiling. Let's take another example where we have heterozygous males and females that will mate. Now we know from Mendelian genetics that the ratio of the offspring will be 1 capital D is to 2 capital D is to 1 small d small d. Now in this case, we would expect that the offspring would have different phenotypes. So all offspring who have capital D would be dextral. Capital D small d would also be dextral coiling, whereas small d would be sinistral coiling. However, in this case, we actually see that all the offspring will show right coiling. And the reason that they will show right coiling is because, once again, pay attention to the genotype of the mother. The genotype of the mother is going to be dextral because it is capital D small d, and hence all the offspring will show right coiling or dextral coiling. So this kind of inheritance is called as maternal effect. So you may be asked questions about this, so please understand how maternal effect works. Now, the genotype of the ovary in which the oocyte develops actually determines the orientation of cleavage, and that is how we can explain why the offspring show a shell coiling structure that is actually dependent on the genotype of the mother.
Scientists have performed many studies to determine how maternal effect occurs. Some genetic studies identified a gene called formin. We now know that maternal formin messenger RNA is deposited in the egg cytoplasm during the process of gametogenesis. Formin is a protein that is able to interact with actin and promotes actin polymerization to form microfilaments. Thus, it helps in aligning the cytoskeleton. Now, the capital D allele of the formin gene gives rise to a functional formin protein. Thus, when the maternal D allele messenger RNA is present in the zygote, it is able to give rise to cleavage patterns that promote right-handed coiling or dextral coiling. The small d allele, on the other hand, gives rise or corresponds to a formin gene that has a frame shift mutation. Thus, the messenger RNA is unable to be translated to give rise to a functional protein. Due to this, the formin messenger RNA is degraded. Thus, embryos that have the maternal D allele deposited in their cytoplasm are unable to form the functional formin protein, and this in turn gives rise to cleavage patterns that correspond to left-handed coiling or sinistral coiling. Now, dextral and sinistral coiling snails develop as mirror images of each other.
Now, if you look at the gene expression patterns in dextral and sinistral embryos, we can actually observe that very early in development, the cleavage patterns are different, and these cleavage patterns influence the location of gene expression in the developing embryo. So if we look at the first figure, the figure has actually been stained with actin, which we can see in the form of these green lines. In the case of dextral embryos that are shown to the right of that blue box, we can actually see how there is curving that occurs, which we do not observe in the sinistral embryos, which is shown to the left in the blue box. And this causes the cleavage to occur in different planes. Now, since the cleavage occurs as mirror images of each other, when we look at the expression of proteins during embryonic development or gene expression during embryonic development, it also shows a mirror image pattern. This is what is shown in the blue box that is in the panel below. In this case, we are looking at the expression of a protein called as nodal. And when we look at nodal expression in sinistral versus dextral, we can see that it is being expressed once again in a mirror image pattern. Nodal is known to cause activation of the expression of Picts. And once again, when we look at the expression of Picts, we can actually see that in the case of sinistral and dextral embryos, they are mirror images of each other. So where Picts is being expressed now towards the left, in dextral coiling, it is expressed towards the right. And thus, all these different patterns of gene expression will result in either an embryo that is going, or either an organism that will show dextral coiling or sinistral coiling. Now let's see what happens if there is an alternate way in which cleavage occurs. Now some mollusks can change the cleavage patterns and that results in alternative body structures. We're going to look at the bivalve mollusk. In the case of the bivalve mollusk, the larvae need to form a bare claw structure that helps them attach to fish and mediates their dispersal in the environment. So when we look at what I mean by this bare claw structure, this is what we can see what is shown in that blue box. Now for this bare claw type structure to form, when we look at what happens during the cleavage process in the bivalve mollusk, we can see the presence of macromeres and micromeres similar to what we saw in snails. When we look at the D macromere and when it undergoes cell division, normally what happens is the 2 capital D will remain as a macromere and we will get a 2 small d micromere. However, the organism has changed the cleavage pattern in such a way that the 2 small d now becomes a macromere, or it is much larger in shape than the 2 capital D. And further development with this larger 2 small d will actually result in this bare claw structure that allows proper disposal of larvae. Thus, alternate cleavage can give different shapes in the organism.
With this, we come to the end of our talk, where we learned about the process of cleavage in snails and the role of foramen and how foramen determines the direction of shell coiling by affecting the process of cleavage.